students. Good afternoon. May we all please settle down as we are about to start in a few minutes. Students, and for those joining us through Facebook Live, a pleasant afternoon to all of you. I am Luisa Falcotelo, your moderator for today's lecture series. So as we start off the lecture series headed by the Department of Biological Sciences, let's have the opening remarks by our Dean of Institute of Arts and Sciences, Dr. Rowena Capulong Reyes. Dean, wala pong audio.
Can you hear me now?
Yes po, you may proceed po.
Dr. Mirna Quinto, Vice President for Academic Development, Mark Isla, Associate Dean of IAS, Benilda Laza, Department Chair of Biological Sciences, Professor Jason Chavez, today's resource speaker, faculty and staff of the Department of Biological Sciences. Dear students, good afternoon. Kamusta kayong lahat? Welcome to today's IAS Vase, otherwise known as the Institute of Arts and Sciences Virtual Activities Supporting Education. This is from the Department of Biological Sciences Research Colloquium Series. How happy we are to be gathered this afternoon. We would have wanted to do this face-to-face, but our virtual presence will make up for it. I enjoin everyone to say hi to each other. Kaya natin gawin yun, kahit na virtual. Today's topic piqued my fancy since we are mentioning sex in the same sentence as a plant. Apart from it appealing to our new plantitas and plantitos, we are touching on the academic side of plants. So I echo the question posed by our scholar today. Sex, location, and ignored features, do they matter? It does, as explained by Dr. Lawrence Kelly of the Graduate School of New York Botanical Garden. He said, and I quote, plants produce 98% of atmospheric oxygen through photosynthesis. Everything that we eat comes directly or indirectly from plants. 1/4 of prescription drugs come directly from plants or are plant derivatives. We know that fossilized plants provide energy in the form of fossil fuels such as oil and coal. So the chosen topic of Professor Chavez really got me reading some more. New scientific research revealed by the University of London in January 2017 revealed that plants with a particular breeding system change their sex depending on how much light they receive. And as such, I'm very keen on finding this out for the plant Antirhea, tama ba yun? Or Guettardeae Rubiaceae. Please correct me on that one, Professor Chavez. I acknowledge the hard work of Professor Benita, who together with her entire faculty lineup, they're busy with preparing for the AUN assessment. We would like to extend our gratitude to soon-to-be Dr. Jason Chavez for giving us time. I know there's a time difference in Germany and the Philippines. Thank you also to our student moderator, Luis Alet, our staff in charge, Binky Borja, technical director, Glenn Boleche, and of course, MCOs Christian Evasco for the technical support. Please, again, allow me to thank everyone who made today happen, faculty and student. Let me end with a quote from Peter Lower. Like people, plants respond to extra attention. I invite you to join us in future webinars. Again, thank you very much.
Thank you so much, Dean Reyes. So participants, let me just remind you of some of the guidelines as we proceed with the webinar. So participants must mute their microphones and turn off their videos as our guest speaker will continue to today for today's lecture. So to formally introduce our guest speaker for today's online series, may we call on Ma'am Benilda Olaza, the Department Chair of the Biological Sciences.
Can you hear me? Can you hear me?
Yes, you may proceed po.
Biology students, faculty, guest participants, Associate Dean Mark, Dean Wen, Bipad, Birna Quinto, and everyone. Good afternoon. I am very pleased to introduce our speaker for this afternoon. He took his Bachelor of Science in Education, major in Biology and Chemistry, as well as his Master's degree in Biology from the University of Santo Tomas. Currently, he is a German Academic Exchange Services Scholar and a doctoral candidate at the Department of Plant Systematics of the University of Bayreuth in Germany. His current research is on the circumscriptions of the Old World dioecious genera of the tribe Guettardeae of the coffee family. And he is also preparing the revision of the tribe's Philippine representatives. He is also a regular full-time faculty of our own department, the Department of Biological Science in FEU Manila, under the Institute of Arts and Sciences. May I present to you our speaker for today, Mr. Jason Chavez. Thank you.
Okay, so good afternoon, everyone. So that's what we say here, Guten Tag.
Good afternoon.
Yes, good afternoon. What I'm going to talk about, so can you now see the PowerPoint presentation? Or no?
Yes, po, sir.
Okay, moment. So when I was asked, like a month ago, what I want to talk or that I'll be giving a talk, and 2 weeks ago, Ms. Borja and Ms. Laza sent me an email. What title should I? What will be my topic? And I was thinking of the title, no. And when I was watching some TFC stuff, I've heard Vice Ganda talking about his younger years, no? It doesn't mean that I'm very old, I'm still young. And he was talking about how people communicated back then. And I realized during my college years, there were no Facebook messengers, there were no Skypes, there were no Vibers, and so what we have then was MIRC, Yahoo Messenger, etc. And for us to know people, we send messages and asking for ASL, age, sex, location. And afterwards, the answer you will be given, you'll be asking for a photo just to look on how they look like, no? So that's why I said, oh, why not sex, location, and features? Do they matter? And yes, they do for this group of plants who have been waiting to be solved for 200 years. So the talk for today, I have decided to give a brief introduction or to recapitulate our basic principles in taxonomy and phylogenetics, also in plant sexuality. Then eventually we will proceed to what I've been working on, a part of what I've been working on, which is on the genus Antirhea. No? So all of us, or most of us, are familiar with the Christian narrative of creation, that there is this omnipotent and benevolent being who created the natural world, or what is surrounding us, for 6 days, and on the seventh day he rested. But there is one part of that narrative that we overlooked, and that is when he presented the organisms, the animals he have created, to Adam. And for Adam to be happy, he asked Adam to give a name to these organisms. And the name that Adam will give is the name of that organism. And all of us are familiar now with the so-called pioneer of the binomial system of nomenclature, and that is Carl Linnaeus. I'm not fond of using the word father of taxonomy, father of genetics, etc., because it's very biased to the male gender. How about the female scientist, female naturalist? No? So Carl Linnaeus, he's a religious person, but he is a walking contradictory. He's quite not humble when he said, Deo creavit Linnaeus disposuit. He thought that if he is the second Adam, which means this Latin statement, it means God created and Linnaeus organized. And scientific names or binomial names are, of course, Latin or sometimes Greek derived. It's beyond the fanciness of these languages. No, we've learned that it is being used because it's the language of the scholars. It is a dead language, but scientific names tell us a story. There are a lot of stories behind it. For example, we have here the fern Gaga marginata, and all of us are familiar with the pop star Lady Gaga, and this plant or this fern was named after her. No, and it's not just that. It's not just because we'll name it after Lady Gaga. No, when scientists were trying to name this fern, they found out that there is this segment or a motif in their DNA, particularly in the MATK region of the chloroplast DNA. And it is a sequence of guanine, adenine, and adenine. So that what made them realize na, pangalanan natin tong Gaga. And there's also one species under the genus Gaga. It was named Gaga monstroparva. I wasn't able to find a colored figure of it because what I've only seen was a herbarium specimen and a botanical illustration. That Gaga monstroparva, the Latin word monstroparva means little monsters. And the authors of that name gave it as a sign or a tribute to the fans of Lady Gaga, which of course we call little monsters. No? Galing na tinatawag yung mga fans niya. Also, you can also, what do you call this? Insult a critique of yours, just like what Carl Linnaeus did to one of his notorious critics, Johann Sigesbeck, a German botanist and a medical doctor. Linnaeus gave the name Sigesbeckia to a weed growing in East Asia. And this weed is foul-smelling, mabaho. So, ininsulto niya doon si Johann Sigesbeck kasi nga kritiko siya. Also, you have si Salpinia pulcherrima, which is in Tagalog called caballero. It was named after Andrea Cesalpino, who published the book or the series of collection De Planti Libri. It accounts medicinal plants. And pulcherrima means beautiful. And who will not recognize SpongeBob SquarePants? And SpongeBob SquarePants is no longer a sponge, but he is a fungus, no, a fungi. When Desjardins, T. and Bruns named a newly discovered sponge in, if I'm not mistaken, in Southeast Asia, they gave it the name Spongiforma squarepantii because it appears like a sponge, but it's a fungus. And when they look at it under scanning electron microscopy, they saw these patterns. And these patterns, they say, it resembles the sky of Bikini Bottom. But the thing is, 2 weeks ago, it was on a Sunday, I was browsing my Facebook. I saw a medical student, no, and shared this meme. I forgot. I think it's from the Chronicles of MD. I'm not bashing someone, no, I'm not criticizing someone. And that Facebook page said, yung BS Bio ka tapos nag-med school ka. And she said, botany, yun daw yung walang silbi sa kanya. And then when I checked the comment section, I kept on seeing taxonomy, no? And I said, really, for sure, is it? It's unbelievable na yun ang sasabihin nila. Bakit? Because how will they know that a particular illness is caused by an organism? What type of organism? No? If the names are not there, they really don't know what they're talking about. For example, you have here Filariasis. No? There are, if I'm not mistaken, there are two types. We have the one that is caused by Buchereria bancrofti and the other Brugia malayi. And these two worms, or filarial worms, are being transmitted by different species or genera of mosquitoes. For Buchereria bancrofti, we have Anopheles, Culex, and Aedes. While for Brugia malayi, the only vector is from the genus Mansonia, no? And also you have here Urticaria, hives. And there are a lot of causes of it, but for some plant species, we have poison oak and poison ivy. And these two names also represent two different species. One is Toxicodendron diversilobum, and the other Toxicodendron radicans. And of course, names can identify what organism can be used for medical purposes. No, for example, Salix alba, from which we extract acetylsalicylic acid or aspirin, but it is now created synthetically. But binomial names or Latinized names, we just don't give it directly. We will follow a particular set of rules. And these rules depend on what organism you are going to describe or what organism you are talking about. We have here the International Code of Nomenclature for Prokaryotes, for animals, the ICZN, for cultivated plants, the ones that we hybridize. We have International Code of Nomenclature for cultivated plants. And of course, we have the ICN, earlier, before it's known as the International Code of Botanical Nomenclature. But it was changed, I think, in 2012 when it was It became International Code of Nomenclature for Algae, Fungi, and Plants. And there's one set of not organisms, but we have here the viruses, the International Code of Virus Classification and Nomenclature. And since we're talking about plants, I'll be talking about plants, I'll be focusing on the ICN. For us to identify if the name of the organism is correct, we have to check if the rules were followed. No? I'll give an example. We have here a species that I described recently, which is Timonius Pesvetarum. By the way, the way I give names to organisms or plant organisms that I discovered, there are only two things. I will name it after a person who provided much contribution to the field, or sometimes it symbolizes something. No? If you will check, I gave Timonius Dumagat to name it after the Agta. And it's not just because of them, but for the habitat of the plant, because it belongs to their ancestral domain, and it is being exploited by private entities, no, and so on. So for Timonius Pesvetarum, it means pes, hope, vitarum, life. So it's a hope for life, for biodiversity, particularly in the island of Palawan, which is the Philippines' last ecological frontier. So in the nomenclatural filter published by Turland in 2019, first is that the name should be effectively published. So it must follow the rules stated in Article 29 to 31 of the International Code of Nomenclature. So this should be published in print or online. So the online publication came into light in January 1, 2012, after the meeting of the International Botanical Congress in Melbourne. And you don't just publish it through the internet or just in magazines or newspapers. This should have an ISBN or ISSN, etc., etc. So next, it must be validly published, and the specimen or the name should be typified by a specimen, not a living material, not those that are cultivated in gardens. You must cut a branch, dry it, mount it, and send it to a herbarium, because that type specimen is the one that will bear the name of the organism. Okay? And next, the name must be legitimate. There are some things about it. We have homonyms, same names, but they're talking about different species. That is wrong. That is a big no-no. We also have tautonyms. Unlike in animals, it is allowed, but for plants, it's not. Example of tautonyms are gecko. Gecko, magkapareho yung genus name at yung specific epithet. Yung gecko in Tagalog, we call it as tuko. No? And of course, priority of publication. Sino ang nauna? So, kung siya ang nauna na na-publish, for example, 2011, tapos may na-publish ng 2017, masusundin dapat yung 2011, yung pangalan ng 2011. Next, phylogenetics. So some biology students are familiar with this illustration or this figure, this drawing. This is found in the notebook of Charles Darwin. It shows the tree of a depiction of an evolutionary tree. But nowadays, due to the lowering cost of DNA sequencing technology, it is being discovered that Darwin might be wrong. It's not a tree. It should be a web, but we have to really discover or explore more, because sometimes the nuclear DNA will give a different relationship compared with the chloroplast DNA and the mitochondrial DNA. That happens with the Rubiaceae, in the coffee family, that the relationships given by the chloroplast DNA sequences is different from the mitochondrial DNA. So how will we reconcile those? Trees can help us with a lot of things. One is that it gave everyone an idea of the classification of organisms. No? Before, when I was in high school, my teacher was talking about 5 kingdoms: Kingdom Monera, Protista, Fungi, Plantae, and Animalia. It was during the year 2000, early 2000. And prior to that, during the time of Linnaeus, there are only 2 kingdoms: Kingdom Animalia and Plantae. And then came Ernst Haeckel, the one who coined the term ecology or ecology. Ernst Haeckel proposed the 3 kingdom scheme: Animalia, Plantae, and Protista. And then came the 5 kingdom scheme, which I have mentioned a while ago, and it was found out again that Kingdom Monera should be divided into 2. So we have the 6 kingdom scheme, and now they are proposing more and more kingdoms. No? So that is one of the things that make systematics very interesting. It changes. No? Well, science changes. And another one, we have, perhaps you are familiar with this, SARS-CoV-2, the one that affected every one of us, COVID-19. And they found out, or they gave a hypothesis, what are the relationship of the viruses, the coronaviruses, that can be found in pangolin and bats. So there is one scenario that perhaps the ancestor of the virus came from bats, then it was transmitted to a pangolin, then to humans, and so on and so forth. And we have to, or next is that phylogenetic groupings. No, some biology students, no, those that are listening, have taken systematic biology or phylogenetics of animals, invertebrates, and you have heard of 3 groupings: monophyletic, paraphyletic, and polyphyletic. And when you say monophyletic groupings, I'll just put this in Tagalog. Ito yung mga grupo na lahat sila ay magkakapatid. Galing sila sa iisang magulang. Okay? Yun ang monophyletic. And when you say paraphyletic, merong magkakapatid sila, pero dun sa pamilya, hindi isinama yung isa pang kapatid. Excuse me for the term. These are the children born out of wedlock. Hindi na isama dun sa pamilya. Okay? Pero anak siya. And the other one, we have polyphyletic. Ito yung mga grupo na iba-iba yung ninuno. Parang yung Brangelina, yung mga anak ni Brad Pitt at ni Angelina Jolie, na iba-iba ang pinanggalingan nila, iba-iba ang ninuno nila, pero nasa isang silang pamilya. So, pero ano ba ang gustong mangyari ng mga phylogenetic systematists, or as what we call them, cladists? We will only use those or we will only recognize those that are monophyletic because those are natural groupings. But what about paraphyletic and polyphyletic groups? So there are two ways. First is to lump, pagsamasamahin mo na lang sila as one entity, or paghiwa-hiwalayin mo sila na iba-iba na sila ng grupo. Okay? I hope you understood it, because later on we will use all these principles to solve the problem of Antirhea. Next, sexuality. In our general botany courses, or in the general botany course, we were taught that plants can be bisexual, having or are hermaphrodites, no? Yung isang bulaklak may lalaki at babae na organ, or pwede din siyang unisexual. Magkahiwalay yung bulaklak ng lalaki o babae. And when I was doing this project, I was talking with my friends here in Germany, Filipino friends, no? And they said, ano ba yung halaman mo? Because I was complaining, I cannot find the character. And they said, I told them, yung ibang halaman, babae. Yung iba, lalaki. Pero yung lalaki, bakit nagde-develop ng fruits? And we know that the fruits are the mature ovary, no? So sa babae yun. So sabi nila, ano ba yung halaman mo? May bakla, may tomboy, etc. So just like humans, plants are also complex when it comes to their sexuality. There are a lot of sexual systems, no? We have monoclynous. These are plants which bear only hermaphrodite flowers, just like your roses, your orchids. And we have diclinous. These are plants. In a single plant, you have male and female flowers. We call them as monoecious, or dioecious if the male and female flowers are born on separate plants. There are also andromonoecious. Just an example of monoecious, by the way, is ficus, yung mga some species of balete, and of course coconut, no? And for dioecious, we have ginkgo biloba, yung for memory, and we have andromonoecious, just like Mangifera indica, your manga, and gynomonoecious, subdioecious, androdioecious, and so on and so forth. So the family, or the genus Antirhea, belongs to the family Rubiaceae. It's the fourth largest flowering plant family, and it is composed of about 14,000 species and 600 genera. And all of its species are found worldwide. There are some records that suggest that it can be found in the Antarctic continent. But majority of the species are found in the tropics, no? And in the Philippines, Alejandro mentioned that it ranks second after the orchid family. And I have here some representative species, no, that can be found in the Philippines. We have santan, Vicia, and Nauclea, or Bangkal in Tagalog, and they say it looks like SARS-CoV-2. Yeah. And how can we recognize Rubiaceae? It's very easy. So I have here a plant, no? It's Vilaria, an endemic genus in the Philippines, and you can see the leaves are opposite, tapatan, no? And if you will look closer, in between those leaves, there is a appendage, no? We call it as the stipules, no? Interpetular stipules. And the flowers of it are said to be have inferior ovaries. But the thing is, the Rubiaceae, large groups of organisms, especially those that are circumscribed with a limited suite of characters, are often problematic when it comes to their taxonomy. And during the past 30 years, Rubiaceae systematis employed the use of the DNA to identify groupings, to identify if this group is natural or not, and so on. But despite of these attempts, these efforts, some groups are still problematic, just like the group that I've chosen, which is the tribe Guettardeae. This tribe is predominantly found in the neotropics, in tropical America. And in the Philippines and in the rest of the world, it is well known because of the beach forest species Guettardeae speciosa. If you go to our beaches in the coastline of the Philippines, you will find this plant. It smells good. It smells like sampaguita. But the thing with this tribe is that since its description by Jossu, by the French botanist Jossu, the generic limits, the boundaries of the genera, the correct name to be used for its species, what characterizes the tribe remains a debate among systematists. That's why in 2010, Charlotte Taylor and Gerald Giroux, when they were doing the nomenclature and the species descriptions of the New World Rubiaceae, they said that the taxonomic stability of the tribe Guettardeae is most likely will not be reached in the imminent future. And in the old world, in the Paleotropics, where the Philippines is situated, majority of Guettardeae species, around 99.7%, if I'm not mistaken, are all dioecious. When you say dioecious again, magkahiwalay yung lalaki at yung babaeng halaman. And these dioecious species can be categorized into 6 genera. We have Antirhea, Bobea, Guettardeae for New Caledonia. We have Hodgkinsonia, Timonius, and Tinadendron. I've been working on 2 of these genera, Antirhea and Timonius, but my work also considered this 4 genera, superficially. But the thing that I'll be talking about for today is for Antirhea. So the genus Antirhea, as currently circumscribed, is composed of 37 species, and in the Philippines we can find 14. 14 were already described, and our country is the primary center of its diversity. So ibig sabihin, halos lahat ng species na na-describe ay sa Pilipinas nakikita. And 13 of these species are endemics. Isa lang yung hindi native, isa lang yung nakikita pa sa ibang lugar, particularly in Borneo, in Sabah. And we can identify them easily, based on their flowers. The corolla lobes, yung margin ng corolla lobes nila are overlapping with each other when it's in bud. So when you cut it in cross section, you will see that the corolla lobes are overlapping. And when you cut the fruits in half, you will see a hard structure a ligneous one, a woody structure known as pyrenes, known as putamen. This putamen was formed by the fusion of the walls of the pyrenes, no? Yun yung naging traditional na definition niya. But the thing is, the classification of the genus Antirhea has been compounded by the wrong, or not wrong, but different views of its sexuality, geographical bias, the limited suite of diagnostic features, and of course, species that were misplaced to this genus. So I will explain this further by going back to its history. No? So the genus Antirhea actually is one of the problematic groups in the coffee family. So it was first described by Josu in 1789. The name Antirhea was given by Josu for the plant Bois de l'Osto collected by Commerson in the island of Réunion in Mauritius. No? And 2 years or 3 years after, Jean-Baptiste de Lamarck questioned the work of Josu. He said, no, it shouldn't be Antirhea. The species of Antirhea should belong to the earlier genus Melanea. Why? It's because Lamarck saw a particular specimen, no? And this specimen was erroneously tagged when it comes to its locality. It was tagged coming from the Andes, but actually the specimen came from Mauritius. So it started to become more complicated. And in 1830, Kandol said that no, it is not Melanea, it is Antirhea. But there's one thing that Kandol said. One species of Antirhea, specifically Antirhea bourbonica, which was during his time known as Antirhea dioica, he said that the flowers of that species are hermaphrodite but abortive, becoming dioecious. No? So, hermaphrodite yung bulaklak, may lalaki at babae, pero eventually, yung babae daw na structure nawawala. Kaya nagkakaroon lang ng lalaki na flower. Then came Hooker in 1873. He said that Antirhea should be represented in the Indo-Pacific region and the Neotropics, the tropical Americas. No? And he attributed to Antirhea several genera, which became the synonyms of Antirhea back then. These are Guettardeae, Titoniotes, and Stenostomum. And after 100 years, Jansen challenged or questioned that very wide circumscription of Antirhea. He said that no, Antirhea should be limited to the Madagascan region, Madagascar and Mauritius. And he said that the species from Madagascar are hermaphrodites. And those that are in the Indo-Pacific region are dioecious. That's why he said, we will resurrect the genus Guettardeae to accommodate species from the Indo-Pacific and to restrict Antirhea to the Madagascan region. But there were two scientists who questioned the work of Jansen. And they said Jansen had a wrong interpretation of sexuality because Antirhea in the Madagascan region are dioecious and not hermaphrodites. and that those coming from the tropical Western Pacific, which Jansen said belongs to Guettardeae, should go back to Antirhea. And this concept of Chow and Darwin is the one that is being accepted now. But there's one problem. The findings or the hypothesis of Chow and Darwin was challenged by molecular data, in particular in the work of the French botanist Achille. Achille and his colleagues used the internal transcribed spacer region of the nuclear DNA. And they were targeting a different genus. They were trying to solve the problem of a different genus of Guettardeae, and that is Guettardeae. But they found out also that Antirhea is not monophyletic. So here, Antirhea is paraphyletic. That's what their findings say. It's paraphyletic. But the thing is, they only sampled 5 species that accounts 10.81% of the total recognized species. And another problem is that the relationships of these branches of the tree are not well supported statistically. It's very low. And that's why they weren't able to provide a resolution of the genera of the tribe Guettardeae. And even the attempt in 2016, there was someone who was working on a particular genus in the tropical Americas. That paper is still not yet published, maybe because of the problematic circumscription of Antirhea. Because Antirhea plays a critical role in the circumscription or in the definition of all the genera of the tribe Guettardeae. Because one way or another, these genera have been related to Antirhea. So can we bridge this question, this problem? The answer is yes. And how I was able to do it. So instead of just using one molecular marker, I utilized 5. 3 from the chloroplast DNA. These are the ATP-BRBCL region. If you took your general botany or in your physiology class, you are familiar with Rubisco, the enzyme Rubisco, the one that is responsible for the dark cycle of photosynthesis. So it's this region, ATP-BRBCL, is the non-coding region situated between the gene ATPB and RBCL. And we have here the ribosomal protein subunit 16 and the TRNT to F region. And for the nuclear DNA, I've used the external transcribed spacer and ITS region. But there's one problem. When I was reconstructing the phylogeny of the group, these markers, which are well known to Rubiaceae systematists to be good in sorting, in resolving relationships at lower taxonomic ranks, seems not to be powerful. And I was having big problems back then because I said I cannot sequence other genetic markers anymore because those that are available in GenBank are very few, and majority are from the works of Achille. It's only ITS. And if I will attempt to sequence everything with other genetic markers, of course, I will try which of these markers or what are the best markers to be used. So it will be very expensive and labor intensive. So when I was looking at the DNA sequence alignments, I saw certain mutations, not substitutions, not transitions or transversion events, But I saw some insertions, simple sequence repeats or tandem repeats. And when I was looking on those other microstructural characters in the genetics, in the sequence data, I've observed that there is this pattern, no? So I employed these mutational characters in the data set also as binary characters, as 0101 and not ATCG, no? And I've employed 3 phylogenetic reconstruction techniques. We have Bayesian inference, maximum likelihood, and maximum parsimony. These 3 methodologies in tree reconstruction employ different types of parameters and calculations. So as a result, what can we find about, what can we see about Antirhea, no? So the sampled Antirhea, we have here 2. Here, we have 1, 2, 2 in this clade, and majority are here. No? We can find that we can find that the Antirhea species are nested in 3 distinct clades. And we can say now that Antirhea is polyphyletic. Balikan ulit natin. Systematis and Cladis say that only groups that are monophyletic should be recognized. But how about Antirhea? Antirhea now is polyphyletic. We have, again, 2 options. It's either to lump everything to include Timonius, Tinadendron, and New Caledonian Guettardeae as one genus, to treat them as one genus. But there's one problem. If we will do this, we will transfer all Timonius species to Antirhea. And Timonius is one of the species-rich genera of the Rubiaceae. It is estimated to be comprised of around 200 to 300 species, no? And it will not be very practical. And aside from that, the only character, morphological character that will unite this single genus, Antirhea, a very broad circumscription of Antirhea, is dioicism. Magkahiwalay ulit yung lalaki at babaeng bulaklak, and the pollen grains. And the characteristic of the pollen grain can only be seen through high or powerful microscopy. So particularly scanning electron microscopy. So we can see a reticulate pattern, no, in the exotesta, or sorry, in the exene of the pollen. So, and these 2 characters that I've mentioned, dioicism and the pollen grains, can also be found in other groups of the tribe Guettardeae. So it is not practical. It will cause a lot of problems. And as scientists, we should give solutions to problems and not create more problems. No? So the attempt now is to split everything. But how? Remember sexuality? As what I mentioned earlier, Candol said that one of the species, Antirhea dioica, which is now Antirhea borbonica, is, the flowers of it are abortive, no? Or dioecious by abortion. So this is the statement, Floribus aborto dioicis, no? And I was very curious about it because when I was checking on the herbarium specimens of Antirhea from the Madagascar region, I was I crushed the anthers, no, the one that contains the pollen grains, no, for supposedly female plants. When I crushed it, I saw that the anthers possess pollen. So I said, kaya nga sinabi nung kabarkada ko, ano ba yung halaman mo? May bakla, may tomboy, and so. And when I was looking on literature, no, it's not on, I found an article published in an ecological journal, luckily. No? Serendipity. Totoo nga yung sinasabi ni Archimedes, Eureka. Chances favor the open mind. That there is this gender variation and primary succession in a tropical woody plant, Antirhea borbonica. So in this clade, no? So their findings said that male plants, male individuals of Antirhea borbonica have the capacity to bear fruits. Nagiging inconstant sila. Nagiging babae sila minsan. Bakit? It's because of an adaptive measure, no, to colonize a habitat. Remember that the island of Reunion is of volcanic origin, no? So para ma-colonize yan, kailangan nila magparami. And to bear fruits, particularly the female plants, they need a lot of resources. And if the habitat is depauperate with these resources, how will they bear fruits, no? Kasi kailangan nila ng mas maraming energy, kailangan nutrients, and so. So sasabihin, yung lalaki na lang ang gagawa din para maparami ang lahi nila. But this lability of male inflorescences cannot be found in these two clades of Antirhea because these two clades are strictly dioecious. And this one are leaky, no? Inconstant. Hindi lang yun dito sa grupo ng Antirhea na to nakikita, itong inconstant males. It can also be found in the genus Timonius, which I am also working on. And these inconstant males can only be found in the Philippines. And when I was reconstructing the phylogeny of Timonius, these Philippine species, which have inconstant male flowers, are positioned before Antirhea 1 clade, no? At yun yung parang sinasabi na missing link. Yun yung hinahanap ng paper na to. Hinahanap nila kung sino yon because it is not known in Guettardeae. And the Philippines will really play a critical role in the evolution of dioecism in this group. No? Kasi yung sa Pilipinas, di ba nga sinabi ko, yung lalaki talaga nagpo-produce ng babae, ay ng bunga. Pero si Antirhea 1, inconstant lang yung kanya. Then, eventually, magiging dioecious na lahat. Strictly dioecious. Next, distribution range. If you will recall the history again of Antirhea, the first attempt was to describe it in Mauritius, no? Then eventually, someone said, oops, let's treat it the tropical Americas to the Indo-Pacific region. And this again was contradicted in 1984. They say, Madagascar region only. And eventually to the Indo-Pacific and again the Madagascar region. But when I was looking again on the phylogenetic tree, it was found out that the species of Antirhea 1 can only be found in the Madagascar region. And those from Antirhea 2 can only be found in the Lesser Sunda Islands, in Papuasia, and in Southwest Pacific Islands, the blue ones, no? While Antirhea 3, this clade, can be found in this entire region. So location does matter. Next, about morphology. Remember, as what I've said, nag-focus yung mga taxonomists noon, hindi sila nag-focus sa sexuality. Nag-focus sila doon sa pyrene, doon sa putamen, doon sa cavity, doon sa locule ng putamen, yung bilang nun. So ilan yung locules na makikita or pyrenes, no? Una, may napansin. So I want you to look on these figures, no? So what can you see on the flowers of these three plants? So the one on the left are males and on the right females. You will see here that the male plants of Antirhea and the female plants, the inflorescence looks the same. The inflorescences look the same. Magkapareho yung bilang ng bulaklak, nung lalaki at nung babae. But for Antirhea 2, mas marami yung bulaklak nung lalaki at pagdating naman dun sa babae, isa lang. And also for Antirhea 3, mas marami yung sa lalaki, mas konti yung sa babae. So that's another set of characters. Another, you have the fruits of Antirhea, of the different Antirhea species. You will find here, remember in your general botany, the part of the fruit, you have your exocarp, yung labas ng fleshy fruit, no? So yung labas niya, we have the mesocarp and the endocarp, which contains the seed, no? The hard part. And here, you will see that the mesocarp of Antirhea 1 is very thin. Actually, nakadikit na nga lang yung exocarp dun sa endocarp mismo. While for Antirhea 2, you will see that it's fibrous, no? Sclerenchymatous. And there are holes on the mesocarp. There are cavities on the mesocarp. No? And these fibers, no, allow the fruit to float on water, no, parang coconut. And madalas nakikita yung mga species ni Antirhea 2 sa tabi ng dagat. And next, Antirhea 3, you will see that the mesocarp here the mesocarp is fleshy, it's juicy, no? So that's another set of characters that can delimit them, no? Hindi yan pinapansin noon ng mga systematists. So what will happen? In this case now, on the results of my study, the three Antirhea clades should be recognized as three different entities. So the first one, Antirhea sensu stricto, this now will is the narrow concept of Antirhea. So this should only be limited to the Madagascar region that exhibit leaky dioicism, monomorphic inflorescences, and thin mesocarp, and locules that are very few, around only two or three. While Antirhea 2 will be treated as a new genus, which will be given a name Achilleanthus, named after Frederick Achille, yung unang nag-attempt to construct the phylogeny of Guettardeae, and anthus means flower, the flower of Achille. And as you can see here, I placed inned, and inned means ineditus. In English, it's in need of publication. No, I'm just waiting for the final result of the review process. No, I have just submitted the revision, no, of the manuscript. So this new genus I have only proposed 7 new combinations because I will leave the treatment for New Caledonia to the authors, the French authors. And of course, all Philippine species now will be treated to the resurrected genus Guettardeae. So 12 species will be going back to Guettardeae, and 17 Antirhea species will be transferred to Guettardeae, giving now the genus Guettardeae with 29 species. No? So what will be next after this? As what I've mentioned a while ago, I'm also working with the genus Timonius, no, 200 to 300. And I have now the results of it, and the manuscript now is to be submitted. I'm just layouting it for the journal, no, and waiting for the publication of the paper for Antirhea. But the thing is, even though I have provided a resolution for Timonius, I have just scratched the surface because this group is very large. It's very big. And I wasn't able to obtain DNA sequences from some Philippine species. No? And next is there are several new species for Guettardeae, Philippine Guettardeae, to be described. And I've been waiting for as what I mentioned, the genus Guettardeae, its resurrection should be published, validly published, and effectively published. And the new species was described by my former students in FEU, some FEU alumni. We're just, again, waiting for the paper for Antirhea. Next is to detect male and female plants molecularly, no? Because it is known that climate change can affect dioecious species. Maybe it can lead dioecious species into extinction. So that's why we need to really know. We are in the race to identify, lalaki ba to o babae? Because it's difficult to identify Guettardeae species without flowers. So we will attempt it using molecular information. And lastly, some ecological studies. I'm planning to do ecological studies to check if environmental factors will affect the development of male and female plants. So that's it. No? Thank you for listening. But before I return the floor to the hosts, I want everyone to remember, especially biology students. I know that some of you will proceed to med school. Most of you will proceed to med school. But don't forget that in your core, you are biologists. And I was frustrated when I see former biology students of FEU talking about some issues, current issues in the Philippines about the environment, particularly the Manila Bay issue. No, that they are still protecting the government because of fanaticism. I don't know if some of you are supportive of this government, but the thing is, we have to look on its impact to our environment because we only have one place to live in. No? And if this is destroyed, and this photo was taken when we were in Mount Iglit, Baco, just to check for the counting of the tama rounds. And when I saw something, and they told us that they burn the habitat just to count. Is it scientifically good or not? Is it good for our environment or not? So Don't forget, you are all biologists. So thank you again.
Ayan. Thank you so much, Sir Jason Chavez, and you truly are a pride of the FEU Biological Sciences. We hope that your research will continue to reach greater heights and that students will be inspired to do the same in the field of research. So to continue today's program, we may ask Sir Jason Chavez again to be ready for the question and answer portion. And now, the floor is now open for questions. Participants who are here in the MS Teams, you may type in your questions in the comment section. And for those who are in Facebook Live, may we ask also to type in your questions in the comment section as well. Our Biological Science Society officers will be gladly to assist you. So, we have questions already for while the talk is ongoing. So, may we call on Ray Matthew James Volante. Please ask your question po.
Hello po. Narinig po.
Yes, I can hear you.
Sir, you have mentioned po kasi that you have constructed trees using for different markers. For example, you have constructed variation inference, maximum likelihood, and etc. So, are there any criterions po ba or standards or any justification po to use a specific method in constructing trees? The best method po in constructing trees po, like for example, when should we use maximum likelihood? When should we use? When is the best time to use Bayesian reference inference po?
Or parsimony. Yan, yung tatlong method. No? Am I right? Which is the best method?
And when's the best time po to actually employ the specific method in constructing trees po?
The specific one. Okay. So first is, actually, I don't want to subscribe to a single method because it's not a one method cures all. No. In the sciences, a scientist should really go, the more evidences you have, the better. So you try to employ these three methods and check if there will be incongruences. If there will be incongruences with the information that they will be giving. Kasi ang Bayesian inference, ang method niyan, iba kay maximum parsimony, iba kay maximum likelihood. Ganito, i-explain natin. The Bayesian inference is more of probabilistic. Probability siya. Okay? Probability siya. Si maximum parsimony at si maximum likelihood, di ba common siya? Kay Bayesian, nakikita mo, I'll share again a slide. Can I share it? Can I? So, I'll share this slide. Para mas malaki. Oh, shoot. Makikita mo dito that in this clade, no, the paleotropical diameter, the first one, the value is 1. This is for Bayesian inference, and you have your 0.74, 0.96, etc., etc. And you have 86 and 88, 94, 92, no? Makikita mo, si Bayesian inference, ang highest value niya is 1. So some journal articles, some statisticians say that it should be 0.95 to 1. Yun ang well-supported. While for maximum parsimony, the bootstrapping method, no, it's hati ang view ng lahat. Some says no, 90 and above. Some says no, 70. It's supported already, no? Just like in my case, I accepted 70 and 70. That's already well supported. Because of the method, kung paano nila sinasampling yon. Yung support ni maximum parsimony and the likelihood, yung bootstrap, depende yon kung ilang beses na nakukuha yung relationship na yon. Kung hindi mo pa, kung if medyo vague pa, di ba naglalagay ka ng replicates, 1,000 replicates, 1,000 bootstrap replicates. Doon ba sa 1,000 na yon, kapag yung 1,000 na yon, lahat ng sampling nun, nakuha yung clade na yon, ang ibibigay na bootstrap, 100. Pag 98% ng sample, nakuha yung clade na yon, 98%. Pag 70, 70%. So, titingnan mo na lang yung magiging relasyon ng dalawa, ay nung tatlong methods. Kung sinabi, 1 si bootstrap, at pagdating ay 1 kay Bayesian inference, posterior probabilities. Then, pagdating kay bootstrapping, 70. There is this manuscript in 1992, if I'm not mistaken. They said that it can be treated as well supported, no? Pero there's one technique para wag ka maging tawag dito time consuming, calculation intensive, kasi di ba siguro yung iba sa inyo ginagamit yung laptop nyo, and to run it sometimes it can take months, just like in my case yung likelihood non neto tsaka parsimony inabot ng 1 buwan mahigit, no? Gumamit muna ako ng isang technique, medyo parang fast track lang muna para makita kung meron ba. So I tried maximum likelihood. Perhaps if you're familiar with RAXML, you can use it. And if you are not familiar with coding, no, kasi command prompt si RAXML, there is this GUI. I don't know what GUI means. Yung parang click ka lang, magagawa mo na siya. You can use it. No. So I hope, and also the relationships that you have will depend on your sampling and the genes or the DNA markers that you are using. And just I want to share something. No, it's a conversation between me and some colleagues that when you run a phylogenetic analysis, hindi mo lang yan isasalang sa computer mo. Sasabihin mo yung software, do this, do that, give the parameters. No. You have to really check the data that you have. You really have to check its quality, if you have done the correct alignments, and so is your DNA sequence good? Because there are some cases I've seen something that the DNA sequences are not good. It's very dirty, but they still used it. I said, unbelievable. How, and the reliability of your result, I don't know. So you really have to check all data. But if you have some data with you, maybe I can help you out. Just send me an email. No? So I hope I answered your question.
Thank you so much po,
Yes,
Sir. So we have another question po, and I think this would be the last. Sir Doc Valdez, are you here po?
Hello.
Hi.
Hi, Jason. Kamusta naman dito sa Germany, no?
Hello po.
So anyway, so you discussed about plant sexuality, right? So in animals, basically most of the process of reproduction is, of course, you have sexual reproduction, no? So in plants, it's very obvious that there is diversity when it comes to the production of plants. Now, what would be the implication of this diversity in plant sexuality in general to the genetic diversity and as well as evolutionary process of most plants. No? Because I'm sure speciation has something to do with sexual reproduction, right? Because a species, you can consider it a species as long as they can propagate, no? So that's why those hybrids, you don't consider them as species basically because they cannot reproduce. So F1 is basically most of the time are sterile, no? But in plants, so like for instance, if you have a hybrid and they can produce an F1 and this F1 becomes viable, so where do you draw the line now of calling it as a new species or is it the same species pa rin ba? You know, I find it quite complicated when it comes to plants.
Yes, that's why you really have to ask the expert for this part for certain groups, no? Because, for example, in the coffee family, it is well known that the coffee family, that species in the coffee family undergo interspecific hybridization. No? And there is one thing in the rules of nomenclature, we use the letter X in the that it means, it's a natural hybrid. A natural hybrid. And it really depends on the expert for that group. And for just as an example, when I was in the Philippines doing field work, I saw an individual occurring in Samar. And in that community, there are two species of Timonius. Timonius finlaysonianus and the recently described Timonius polychlorus. And when I was looking at this individual, I said, no, it's in between the two. And I said, I cannot describe it as new one, but I'll just keep there first. And I need more evidence if this really is a natural hybrid, or will this propagate or no, naturally.
Exactly, no. It has also implication when it comes to species identification, no. So that becomes a problem then. No? So do you think a use of molecular markers would be ideal for it to be used as a basis for speciation of this hybrid?
Maybe we can look at it because there is one case, no. It is not unknown to my colleagues in the Philippines that I was having problems with one genetic marker that I've used. And
especially the DNA barcodes marker.
Yes. What happened is that in this marker, supposedly, this gene is only a single copy. But the problem now, there are different copies. There are 3 or 2 copies of this particular gene in this group of plants. So maybe
polymorphic.
Yes, it's polymorphic. But when I was also looking at the DNA sequences that was obtained from these three bands, no, there are certain changes. There are certain what do you call this? Certain
variation.
Yes.
Pero same gene.
Same gene. So, hindi namin alam kung saan to nanggaling. And that's one of the things that I really want to explore, but it needs a lot of, funding for that. And fresh, good samples.
Ang hirap naman ng halaman, kayo nga lang siya. Anyway, that's all Jason. Okay na ka namin makikita.
That's why when people in the Rubiaceae, particularly there's one foreign systematist, no? When he learned that I'll be working with this group, ang sabi niya kagad sa akin noon, oh, you're going to work with garbage, which is really garbage. Naubos ang buhok ko dahil dyan, dahil sa grupong to.
Well, it's a challenge, you know.
It's a challenge, and I can
Made
say,
it.
yes, I was able to resolve a problem that has been disputed for how many years. Yes.
Anyway, thank you. Good luck.
Ayan, thank you so much.
Again, with questions?
Ayan, participants, if you still have questions, we will consolidate questions to be answered by Sir Jason. So due to time constraint, we'll continue with the program. So now, ayan, so thank you, participants. So let's proceed with the awarding of the certificate. So this certificate of appreciation is given to Sir Jason Chavez for his valuable contribution as speaker during the Biological Sciences Online Lecture Series 2020 with the theme Sex, Location, and Ignored Features, Do They Matter? Solving the Centuries-Old Puzzle of Antirhea. Held on 10th day of October 2020, signed by Ma'am Benilda Olaza, Department Chair of the Biological Sciences, and Dr. Rowena Capulong Reyes, Dean of the Institute of Arts and Sciences. So participants and guests, may we please ask you to please open your cameras and let's take a picture with our guest speaker.
Thank you, Sir Jason.
No problem.
See you next
Thank you,
year.
sir.
Pero for better clarification, ask nyo na lang din. You wait for the update of the cell development.
Okay, Arlene will take the picture. Arlette.
Are we all ready na po?
Sir Jason.
So, smile everyone. Okay, one, two, three. Okay na po.
Thank you.
Thank you so much po, Sir Jason Chavez. So to formally end this series, may we call on the Associate Dean of the Institute of Arts and Sciences, Sir Mark Isla, to give the closing remarks.
Thank you very much. Can you hear me?
Yes po.
All right. To our Dean, Dr. Rowena Capulong Reyes, our guest speaker, Mr. Jason Chavez, and to Ms. Benilda Laza, the Department Chair of the Department of Biological Sciences, faculty members, Dr. Marcos Valdez, students, good afternoon. First, I'd like to congratulate the Department of Biological Sciences for successfully organizing this lecture series, which is part of the Institute's Virtual Activities Supporting Education, or Vase. I'd also like to sincerely thank Professor Jason Chavez of our Department of Biological Sciences for generously giving us his time and for sharing his research on solving the century-old problem of Antirhea. I may not be able to relate to the study that has been presented, but I hope that the study will inspire you to will inspire more future biology students to continue to engage in research related to systematics and other fields of biology, so that there will be more scientists who can provide knowledge and scientific evidence from the diverse flora and fauna of our country. The Department of Biological Sciences boasts a number of researchers, also faculty members, who are well known in the different fields of biology, and I'm confident that the students of biology would be able to gain quality education in our university. Rest assured that the Institute and the Department of Biological Sciences will continue to strive to provide students and other stakeholders programs and activities that will present studies discussing issues and trends that can be useful in our understanding about the concepts that you can encounter in your classrooms or courses. So thank you for your active engagement with our speaker and for raising some of important questions. And I hope that you're able to learn a lot in today's lecture. So stay safe and have a good day.
So thank you so much, Sir Mark Isla. So evaluation form is already posted in the comment section for biology students. We also sent the link in your respective Facebook group chats, your batch GCs. May we ask you to kindly fill them up, and e-certificates will be given to the participants afterwards. So that concludes today's online lecture series. We appreciate the participation of Dean Reyes, Sir Mark Isla, the Department of Biological Sciences professors, and the biology students. We hope that you've learned a lot throughout the lecture series, and good luck, students, to our upcoming midterms. Stay safe, everyone, and have a productive day. Good afternoon.
Thank you also to Sir Christian Evasco. Thank you.